Hello, these are your first set of metabolism and enzyme notes, and we're going to start on this side of the big sheet where it says metabolism at the top. So metabolism is just a fancy way of saying all of the chemical reactions in your body. Remember, we're made of cells. Cells are basically just bags of chemistry. You are basically just one walking, eating, breathing bag of chemistry. And when we talk about metabolism, we're talking about the sum of all of that chemistry, right? And usually, when we talk about metabolism, we are talking about energy, right? So energy is not this kind of mystical, kind of poofy, weird thing that just kind of comes and goes. I want you to start think of it as the movement of electrons. That's really what energy is. So electron movement. Right, electricity is the movement of electrons, and that's what a lot of us would think about when we think about energy. So start thinking of energy as something a little more concrete than just something that's just kind of there. It's actually the movement of electrons. And there are two basic types of metabolism and using energy that we're going to go over. The first is catabolism, and the second is anabolism. So catabolism is, scroll down. Catabolism is when you break a chemical bond. You're going to break a chemical bond, and as you break a chemical bond, you are going to release energy. You're going to release energy. So backing up just a second, remember that bonds are just the sharing or transferring, I'm going to abbreviate transferring, of electrons. The E with the negative sign is an electron, and again, they have a negative charge, so hence the negative sign. So that's shorthand for electron. And when you release energy, what you're really doing is you are displacing those electrons. You are displacing those electrons. This is going to get you what's called an exergonic reaction. And in chemistry, you didn't call it exergonic, you called it exothermic, but it means the same thing. Oops, exothermic. Thermic is just for heat energy, and gonic is total energy, but you can think of them as the same thing. And what you're going to calculate when you do stuff like this is something called delta G, and that's free, what's called free, quote unquote, energy. So let's take a look at what this looks like. All right. So you're going to start over here with a substrate, and that substrate has some amount of energy, right? It's going to run through a reaction, and when it gets to its products, if it's exergonic, if it's a catabolic reaction where we're breaking bonds, the products have a lower energy than what the substrate started with, right? All of this energy has been lost. It's somewhere else, right? It's not with the products anymore, so it went somewhere, right? So this is called a negative change in free energy, a negative delta G. We have lost free energy, right? One reaction where this happens, again, we're going to be breaking things in a reaction like this, is cellular respiration, right? So in cellular respiration, you start with glucose. Glucose is a high energy molecule with a lot of bonds, right? Just like our high energy substrate up here in the graph, right? We're going to essentially burn it with oxygen. I'm going to go ahead and just write high energy here. High energy. And our products are carbon dioxide and water. These are inorganic. They don't have a lot of carbon, hydrogen bonds at all, actually. Inorganic, I want to say low energy, which is characteristic of our products. So here we have glucose, a high energy substrate, and we end up with CO2 and water, low energy products. So this is a catabolic reaction. We've broken glucose and we've gotten lower energy products. And the energy, this lost energy, this negative delta G, is the energy that we get from cell respiration. And this is what ATP is. That is what ATP is. That's what the mitochondria are going to produce. So let's look at the other side. An anabolic reaction, or an anabolism, is going to build bonds. It requires energy and is called endergonic. This is the same as endothermic. Remember, thermic is specifically heat energy. Endergonic is like total energy, but for us, we can think of them as the same thing. And we're going to get a positive delta G. We're going to have products that are higher energy than our substrates. All right, and that's what we have here. We have a lower energy substrate in relation to the products. In this case, this is the amount of energy that the products after the reaction have gained. So that is a positive delta G. Right? They've gained energy. Right? And if you're kind of like, well, where'd that energy come from? Well, we'll get there. It doesn't just come out of nowhere. We'll talk about it. So an example of this is photosynthesis. Right here, you are building something rather than breaking something. So here we start with the same, actually over here, we have the carbon dioxide and the water again. Remember, those are low energy inorganic molecules. And we're going to add energy. This is our added energy. And we're going to get a higher energy product. So lower energy substrates, higher energy product, right? We've built something here, right? And what you're going to start to notice is that every time in biology you need to build something, you're going to have to break something to get energy, right? You have to build something, you have to break something. You have to build something, you have to break something. And that's because building things requires the energy that you get from breaking something. So it's kind of like Wreck-It Ralph. Wreck-It Ralph breaks stuff and Fix-It Felix fixes things. So Wreck-It Ralph is kind of like the catabolistic reaction, and Fix-It Felix is like an anabolistic reaction or an anabolism. But if you stop breaking things, if Wreck-It Ralph quits, then Fix-It Felix is kind of out of a job as well, right? So these two reactions are dependent on each other. They're what are called coupled. So this is what's called energy coupling. There's a cute little diagram down here, but the gist of this is, I'm going to go ahead and type this, is that you break something to get energy and then use that energy to build something. And this is exactly what your cells do. So I'll give you a quick example. All of the food you eat, right? So I'll say food eaten gets digested into monomers, right, by your digestive system. So digested into monomers, and this is a catabolic reaction. And then once you've digested those monomers and they're distributed, throughout your bloodstream to your cells have to build your molecules. So then you construct, then you build things like proteins, lipids, nucleic acids, right? This is anabolism. Where did the energy for this anabolism come from? Right? Where does that ATP come from? Because building molecules is expensive, right? Well, it comes from the energy right up here in cellular respiration. It comes from burning glucose, right? Carbohydrates, this is why carbs are associated with quick energy. It's because it's what your mitochondria prefers to use. Carbohydrates provide you with a high energy molecule that after a catabolism, a catabolic reaction, releases a lot of ATP. That ATP can then be used by your cells in an anabolism, or an anabolic reaction, to then build proteins, lipids, nucleic acids, and anything else that it needs. So these two processes are very, very closely related to each other, right? So right over here, I'm just going to say, what are we breaking? We are breaking, oops, that should be a B. We are breaking glucose to get ATP. And then we are using that ATP to build all the molecules you need. So I'm just going to type that last one. I'll say all the molecules you need. So let's go over to the other side. Oops. I want the side with the ATP. Here we go. This side. All right, let's talk about ATP in a little bit more detail, because that's where we're headed. So ATP, which you've learned about when you were freshman in freshman bio, you've probably forgotten most of it, and that's okay. We're going to go over it right now. Is actually a modified nucleotide. What you're going to see in biology all the time is that you don't have to reinvent the wheel. Evolution will favor modifying structures rather than building them up from scratch. So ATP, a cell's energy molecule, is actually simply a modified nucleotide. Here we've got adenine, which is a base. So you've got A, T, G, C, and U, uracil, right? So here we have adenine in particular. Ribose is our sugar, and we have 3 phosphates. Tri means 3, right? Adenosine triphosphate. Adenosine triphosphate. And again, if you don't have these 2 phosphates, it's just a nucleotide, right? So I'm going to zoom in here. It's worth remembering that phosphate is a PO4. It's not just a phosphorus, it's a PO4, and it does have a negative charge. And if you look at ATP, adenosine triphosphate, all 3 of these are going to be packed together, right? You're going to have all 3 phosphates on the same molecule. If you have just one of them, so as it's drawn right now, you really just have one attached and the other 2 are kind of floating over here, right? When you have one phosphate attached, this is called AMP, adenosine monophosphate. When you've attached a second one, now you've got 2 attached, it's called adenosine diphosphate, right? And when you've attached all 3, it's called adenosine triphosphate, right? Also, for future, you don't actually have to draw all of these structures. You can just do a little symbol, you can just do like a little square or a rectangle here. These are just kind of visual models to help us communicate a topic, and it's whenever we simplify things, it's just easier to go about it. So we're just going to simplify this whole adenine ribose structure as a box, right? So over here, if you wanted, and you wanted to draw ADP, you can just go ahead and draw the box and then the 2 phosphate groups, right? Over further, if you wanted to draw AMP, you can just do the same thing, but with, just the 1 phosphate group on it, right? But the reason why this is such a good energy molecule is because you're packing negative charges together against their will. Remember, it's kind of like taking 2 negative sides of a magnet and trying to force them together. They're not going to want to do it, and they're going to repel each other just because of the physics of negative charges, right? So packing these molecules together gets increasingly difficult, right? This first phosphate group, I'm going to call this phosphate group number 1, is here. It was there first, and the second group is kind of invading its space. So when an enzyme is shoving these 2 phosphates together, this bond right here is pretty high energy. It's not the highest energy, but it's pretty high. It's stored. We're storing energy when we make this bond. right? Anytime you make a bond in biology, you're always going to store energy, right? And that's a difficult enough bond to make in and of itself. But now, when we add phosphate number 3 on, now we're pushing a third negative charge into 2 other negative charges. So what was a difficult task becomes even more difficult. But because this is so hard to do and requires a lot of energy that's being stored in these bonds, it makes this molecule an excellent energy storage molecule. And in particular, this third bond, because it's so difficult to make, is the highest energy bond in this molecule. Highest energy bond in the whole ATP molecule. Think of it like spring loading something, right? You have a spring and you're pushing it harder and harder in, and it could like release and like fling something out at any moment. It's like stuffing a briefcase. You stuff it, stuff. And at the very last second, you're able to zipper it all up. But as soon as you undo the zipper, the whole thing's going to pop out and all your clothes go everywhere. That's kind of like what ATP is. It's a spring-loaded molecule ready to release that energy. And it's spring-loaded because you've packed all these negative charges together. And they really don't want to be together. All right, so let's talk about ATP in action, right? So what does ATP actually do with all of its newly found energy? Well, it's going to do something called phosphorylating. And this is called phosphorylation. When you say a molecule has been phosphorylated, you're basically saying that it's had a phosphate group added to it. Phosphorylation is the action of phosphorylating. It's the verb. Right. And phosphorylating, once you've received a phosphate, it makes you reactive. Makes you reactive. So this is going to activate the molecule. Whatever molecule receives that phosphate group, you're going to get activated. Another way to say this is you're reactive, and another way to say this is that it causes a conformational change, a literal shape change in whatever molecule is getting this phosphate group. So we can just go ahead and add here that we are adding a phosphate group, and as a result, we're activating it. Whatever receives it gets activated. And the net outcome is that we're going to do work. Things will happen. Something is going to happen, and we're going to talk about that something here. So here's an example. Here we've got a molecule, and we can go ahead and just call this a protein because most of the time things that get phosphorylated are proteins. So we can just go ahead and do that. This molecule right now is inert. It's not really doing anything. It's lazy. It's hanging out in the cell doing nothing. But it could if it only had a phosphate group, right? So ATP is in your cells. It's constantly being manufactured by the mitochondria, right? And it diffuses around in the cytoplasm. ATP is going to phosphorylate this lazy molecule with help. And the helper here is kinase, right? Kinesthesiology or kinetic energy is like the study of motion or movement of molecules. So this thing is going to make this lazy molecule move, basically. And it's going to do that by grabbing a phosphate and sticking it on to this, this protein, right? So this kinase, and all kinases will do this. Is it going to let me type that? I'll just handwrite it. It's going to take a phosphate group from ATP and attach it to another molecule, whatever that molecule is, another molecule. That's what kinases do. So this kinase rips off this third phosphate. You've broken this bond over here, right? And in doing so, you release quite a bit of energy. And what you're left over with now is ADP, right? Because that third phosphate has been removed. and it's been stuck, it's been attached to this molecule, right, our protein molecule. And what happens, remember we talked about what happens, whenever you do this, oops, whenever you do this, you are going to activate. You're going to make this molecule reactive. You're going to cause a conformational change. So this is why it looks a little bit different. We've caused a shape change in this molecule. And that means this molecule is active. It's ready to do work. It's ready to do something, right? There is a shape change. Do delta for change, shape change, which means this is a functional active molecule. Remember, everything in biology is based on shape. The shape of an active site to fit a substrate, the shape of the receptor for insulin, everything is going to be based off of shape for the most part. So the fact that this shape change has happened is the most important thing here. It's like throwing, and remember, these proteins have complex structure. They have secondary and tertiary folding with all their hydrogen bonds and R group interactions, and you've kind of thrust this massive negative phosphate with its full-on negative charge into the middle of this protein, and it's going to kind of freak the protein out, and it's going to have a shape change, right? It's going to push some of the other negative charges out of the way, and you get a shape change. But it does make this molecule active, right? And it can do work. It can do its job, whatever its job is. Maybe it is to catalyze a reaction. Maybe it's to partake in a series of messenger sequences that are going to trigger something else in the cell, but it's going to do something. That's the point, right? Now that we've done something, we have to restore essentially homeostasis, right? You have to recharge ADP. Think of ADP as like a phone that you've used all day long and it has no more battery charge left. You've got to plug it back in. So we have to reconnect a phosphate onto ADP to make it a fully charged ATP that can be reused. Similar thing with this protein. It's great that it's been phosphorylated and then it's active, but you have to inactivate it so that it can be active again, right? So you have to kind of restore the baseline. And that's going to happen due to phosphatase. Phosphatase, just like kinase, is an enzyme, but its job is to pluck off the phosphate that's stimulating this protein or this molecule, right? So this is going to, I think I can type over here. Type over here. This is going to remove the PO4 from the target molecule, and it's going to inactivate it. It inactivates it. So what you're left with is the original molecule inactive with this phosphate group, right? Because this is all happening in the cell, you are going to build a higher concentration of both ADP and free phosphate groups in the cytoplasm, and they will diffuse. They will diffuse from a high concentration in the cytoplasm to a lower concentration in the mitochondria. Remember, the mitochondria is making ATP, but it's taking in ADP and phosphate because it's diffusing in from the cytoplasm. So these two substrates, which we need to put them back together, we have to make this bond. And that bond is going to take energy, so we're going to talk about where it comes from, but they're diffusing back into this into the mitochondria. Here we are. Oh, I didn't want to draw that. Sorry. Deal with it, it's okay. They're going to diffuse back into the mitochondria, and there's an enzyme in here, right there, called ATP synthase. Here it is, ATP synthase. We'll do details of this later, but for right now, just know there's an enzyme in there that is going to take ADP and phosphorus, or a phosphate group, and it's going to reattach the phosphate group onto ADP to restore ATP. It's like plugging in your cell phone at night, and we've fully recharged ATP, and we can reuse it, right? So that is what ATP synthase does. This is an anabolism. This is an endergonic reaction because we are building bonds, right? We're putting substrates or reactants kind of together, and we're making a higher energy product. Just like we talked about before with Wreck-It Ralph, though, this is an energy-intensive process, right? You've got to get this energy from somewhere. So how do you fuel this anabolism endergonic reaction? Well, we've kind of already talked about it. You burn glucose. You burn glucose. You have a high energy glucose molecule that you break into lower energy products. And in doing so, you release ATP, or you synthesize ATP. This energy is the source of the energy, at least some of it, because your body needs some of it. So I'll say some of that energy is used for this ATP synthase over here, right? So every time you need to build something, you've got to break something. So here's your accompanying catabolism exergonic reaction that fuels your constructive anabolistic endergonic reaction over here. So I don't have things to demo this with, but I do hope this helped, and we will see this in more detail. I know there's a lot in here. Don't get overwhelmed. We will revisit this. We will spend plenty of days. We'll spend an entire week talking about this process in the mitochondria, and I will see you next time.